for my ID table, I wanted to do share iNaturalist, and I realized I've been making slides by taking like screenshots of iNaturalist. I'm like, why am I doing that? Because iNaturalist is like a dynamic living breathing website, and it is better for me to show it to you. So I have gone through and looked at what is in our project, and I want to highlight some really neat finds that our members have had, mostly in April or a little bit in the beginning of May. The other thing I wanted to show you was something kind of cool. I was excited to be invited to do. Now, iNaturalist obviously is a huge repository, has a lot of cool people with little niche areas of expertise, and I got these invitations, which I was super excited about. They're doing these kind of experiments, and the purpose of the experiments are to sort of estimate the accuracy of iNaturalist observations. And I believe that they're also trying to figure out, what qualities are associated with a high level of accuracy. So you all may, those who have iNat and who iNat regularly know, something will become research grade basically if it has like two concurring IDs, but obviously you can find two people to agree to a lot of things that aren't necessarily correct. So, I think what they're trying to do is figure out what are the signs that that an observation is, really accurate, that an identification is accurate or high quality. So I did the first one in March, and it gave me 9 observations to go through, and it was cool because basically it wasn't just that I had to say what I thought they were. Which I guess I could show you. And some of them, I was like, I definitely wouldn't be able to tell you what this is. As you can see, mine are almost all boletes. That's like my area of interest. And some of them I was like, there's no way anyone could tell you what this is from these pictures. And some of them I was like, I know exactly what this is. So I was really excited to participate in that. And I haven't looked at the one I just got from today, but I guess it's another round of it. In other bolete news, there's a new edition of the Northeastern Bolete book by Alan and Arlene Bassett, and it's just come out. They're going to be 2 of our guest mycologists at Sequanota this year, which is awesome. They'll be giving a bolete workshop. So I highly recommend that everyone come to Sequanota because they're incredible experts, not just on boletes actually, but on mushrooms all over, really all over America, but particularly in the East Coast. And they're wonderful people. If you've had a chance to meet them before, they are just prolific scholars and authors and really lovely human beings. So that's going to be a huge highlight. So plug for Sequanota, plug for boletes, plug for iNaturalist, and cool experiments. Now back to the topic at hand, I'm going to go to our project. So just to remind you all, when you go to the iNaturalist website, you have up here a search button. And if you want to find our project, you just start typing mycological association. All I have to type is mycological. I get our project at the tippy top. If I hit view observations, I would just go to a list of observations, so I wouldn't get to see the neat dashboard of our project. So instead, I'm clicking on about, and there's our beautiful project. It's a big project. So as you can see, we have 235 members in our project, and we collect basically any of those members' observations that are made in like the DMV area and parts of the Eastern Shore. So I want to show you a list here of our most recent observations. And what I thought I would do, well, let's see. I guess the first thing, let me go to the project overview, go to view all recent observations. And I saw people had done made some really neat observations, but I wanted to show you how you could like kind of zoom into what you're interested in. Well, first, just looking at the list, I see some really neat rotten orange with some cool penicillium that Elizabeth and Matt posted. That's really neat. It's called Penicillium digitatum, or however you say it, because when you look at it under a microscope, it has these like finger-like projections that are really cool. And anyway, that's a neat picture. But I wanted to show you how to filter. So basically, I'm putting on a filter just for, let's do a date range from our last meeting to this meeting. So Tuesday, April 2nd was our last meeting. Today is May 7th. So, and then I'm just going to update the search. So we've had 650 about observations, almost 200 species, probably more because probably under-identified. And I thought it would be fun to look at some of the ascomycetes, and particularly some of the higher ascomycetes, the Pizizomycetina, Pizizomycetina, which includes some of these cute little cup fungi, but there were a few that I really wanted to show you that I thought were special. Let's see. Sorry for the scrolling. I know not everyone likes that, but this one is Isabella and Jeff both found, and I actually only this one only came to my awareness last year when I saw someone else post it. It's a cool erumpent fungus that grows on holly leaves, and it's called Macroderma curtisii. So I've been looking for it on holly leaves, but I haven't been able to find it. But it's like almost a little kind of cup that opens up. It starts off closed and then it opens up. And I thought they took some really nice pictures of it. You can see they had multiple observations. So good job, Isabella and Jeff. You kind of see why they call it erumpent, right? And you see the frayed edges. Hopefully you can see how it's zoomed in. So it's like, it's almost like, and here you can see tiny ones. It starts off almost like a little bump and then it opens up and you can see the kind of cup-like inside, and that's the fertile surface. So really neat find. This is fun too. Lophodermium pinastri. This is like a Lophodermium is like a ascomycete that I think there's a lot of different species and there seem to be very specific to their host plants. But this is one that grows on pine needles. And so it's fun if you're ever looking in like a big, they just look like dots on pine needles. But if you're ever looking at a big pile of pine needles, look for this guy. But getting to the sort of more big, impressive ascomycetes, this is something I've never found. And I would really love to find it. It's a swamp beacon.